Hi, I'm Heena Modi of PlantShift, and this is Professor Geoffrey Pilkington. He works at the University of Portsmouth. He's been kind enough to take some time out to talk to me about his role at the university. So Geoffrey, do you mind explaining what you do there?
Yes, so I'm Professor of Cellular and Molecular Neuro-oncology at the Brain Tumour Research Centre at Portsmouth. I've been working in neuro-oncology for a very long time. This is brain tumour research in its simplistic term. I think I started in the very early 1970s, and I ended up as Professor of Experimental Neuro-oncology at King's in London, and then moved down to Portsmouth about 13 years ago. So we've set up a brain tumour research centre. We've gone from having 4 people to 24, 25 people there.
Wow.
And we have 5 research subgroups there, looking at diverse areas of research, including looking at the blood-brain barrier, looking at the tumour microenvironment, looking at mitochondria, the energy metabolizing centres, the batteries of cells, and looking at pediatrics. And the 5th group is therapeutics, where we're looking at both new generations of drugs as well as repurposed or reformulated drugs which may be useful to treat brain tumours with. So that's what the centre's remit is there. We've just finished a 5-year cycle of core funding from a charity called Brain Tumour Research, and that's enabled us to bring in funding from other bodies to supplement the team with postdoctoral workers and PhD students. And this is very welcome because the amount of money that's available for brain tumour research throughout the United Kingdom has been dreadfully low over the last few decades.
Okay.
So what we have is a situation where people who come into the lab as young researchers are not able to further their careers because the rungs in the funding ladder are missing, and they invariably have to leave and go and work on perhaps breast cancer or some other branch of neurosciences, or perhaps even to leave the discipline altogether.
Shame. In terms of your findings, I mean, you've been working on it several amount of years, but what have you found so far?
So we've looked at a number of drugs to show these to be effective against brain tumours, and we're trying desperately to get those into clinical trials at the moment. We've also found that the mitochondria, these are little bodies which produce energy for the cell. They have their own DNA as well as the nucleus of the cell, and we're screening for DNA mutations within the mitochondria, and we're able to use those to inform about drug sensitivity and be able to eventually stratify patients into those who will get different forms of drug for the same disease. There are a lot of methods like that which are being used. We're particularly interested in some of the pediatric brain tumours because some of those are eminently curable, but they go that's at a cost of the quality of life because the toxicity of the drugs is quite bad. So we're now again trying to stratify those patients and to treat them more appropriately. Perhaps the most exciting of these areas that we're working on is developing 3-dimensional models for brain tumour invasion, how the primary brain tumours, the tumours which start from the substance of the brain, can diffusely invade into the adjacent normal brain. We can investigate that in 3-dimensional models. We can also model the blood-brain barrier. The blood-brain barrier is very important because that prevents certain agents which might have therapeutic potential from actually getting into the brain. So if we can model that, that will enable us to get very important information in key pathways that are involved there and help us to do 2 things. It'll help us, 1, to get drugs that wouldn't otherwise cross the blood-brain barrier in there, and it'll also help us to see what happens when breast and lung and skin cancers are spreading into the brain and perhaps stop that passage of cells into the brain itself. And all that will be done with the absence of using in vivo animal models.
Jeff, I hear that there are graduates that are beginning their career with you and basically not experimenting on animals. That sounds amazing. Can you tell me a bit about that?
Yes, so we have quite a few PhD students. I think we have about 9 PhD students in the lab at the moment. 2 of them have just graduated, but we'll be replacing them in October.
Okay.
We also have a number of Erasmus students who come in to do research internships from all over Europe, and we have BSc and MSc students who do projects in the lab. But more recently we've been able to secure some very welcome grant support of students who are in the 2nd year, just finishing the 2nd year of their first degrees, and this empowers them to come into the lab for periods of 8 weeks to do work on non-human research, and this is support from the Dr. Hadwin Trust. So this is very useful in getting young people involved in these three-dimensional assays that we're developing and getting involved with non-animal research within the laboratories. It's a new way forward, I think. Many of the people who are interested in this want to do their final year projects in the lab, so they complete their summer studentship where they've worked 8 weeks in the lab, and then they come back the following year and capitalise on that project and then produce a very good piece of work which we hope we can then submit for publication or presentation at a national or international meeting.
Excellent. You're quite pro finding an alternative to testing on non-human animals. Why is that? Why do you think it's so important?
I think there are reasons why some of the animal models are not appropriate for studying the human situation. So we're trying to model using all human cells cultured under human serum conditions and so on. The antigenic nature of this, the brain is different when we compare animals with humans. There are reasons why people would want to use animals in terms of treating humans. And the potential toxicity, but many of the agents that we're using at the moment have already been used for clinical applications, so they've already gone through those toxicity tests, but there's still demands to run those through animal models. And I think what we can do is, because we know about the toxicity, we can then simply fast track those through our three-dimensional human models and get them straight into clinic, which not only reduces the number of animals that are being used in the lab, but also provides a means of fast-tracking them into clinical benefit for patients who are suffering from these life-threatening diseases such as primary brain tumours.
Perfect. Thank you, and good luck with your research.
Thank you.